And Professor Zhao, I just want to mention that we got more than 500 registration and we had to actually stop it at 500 because we cannot take more than 500 people for our webinar. So you can imagine this is probably the highest, popular talk like what we are having like in ISA. So we are extremely grateful that you are here. Um, just 1 minute about the ISA. It's International Indian Statistical Association. Many of the people on the line, they might know about this association. It's definitely like, a global association, whereas we give some emphasis like on the Indian subcontinent, and we foster collaboration, research, and like, we try to promote new career people, through like any of these our platforms, whether it's webinar and some other activities we do. And also we bring people like you, Professor Chow, so that like our community can learn more about like the cutting edge research, what is happening in the field of statistics and data science. So saying that, I just want to introduce you. So Professor Chow is an associate professor of biomedical data science, computer science, and electrical engineering at Stanford University. He works on developing cutting edge AI for biomedical applications. His group developed many widely used innovation, including EcoNet AI, Gradio, and SynthMOL. He has received a Sloan Fellowship and Overton Prize and an NSF Career Award and 2 Chen Zuckerberg Investigator Awards, and a top 10 clinical achievement awards and several outstanding paper awards at different AI conferences and faculty awards from Google, Amazon, Adobe, and Apple. So, I just read, and it's like, people can already understand, that how much like, grateful we are that you are here with us. And today you're going to talk about the virtual lab of AI scientist agents. So I'm not taking any more time. So over to you, Professor Chow, and really welcome to this ISA webinar.
Great. Thank you so much for the welcome, for the introduction, and really nice to meet everyone here. Yeah, so looking forward to having a very lively discussion. So happy to take questions throughout the talk. So don't have to wait until the end. Yeah, so what I want to do today is to discuss and share some of the recent research we're doing around building AI scientist agents. So these are basically agents, AI systems that we built on top of large language models, where we enable language models to access different scientific tools. And we also specialize these models with additional training to become better scientists. So I'm quite excited about these ideas of AI scientist agents, and I want to give a few examples to illustrate what we can do with these AI scientist agents for both like drug discovery efforts in the first half and also for data science in the second half. So in the recent work, we have developed this sort of platform that we call the virtual lab, which is basically a platform where we have teams of different AI scientist agents that work together. So I want to spend maybe the first half of the presentation describing a bit of this, introducing the virtual lab platform. So the motivation for this virtual lab is that, so basically we wanted to create these teams of AI agents to mirror my physical lab at Stanford, right? Because as a professor, one challenge is that we have a lot of projects that I have time to do, right? Maybe I have like hundreds of different potential collaborations, but only have time to do a couple of these. So we thought a couple of years ago that, oh, wouldn't it be great if I could actually create teams of AI agents to sort of mimic my physical lab, so then I can have the AI agents work on all these projects that I don't have the time to do myself. So that was sort of the original motivation. So we created this virtual lab to sort of mirror my physical lab. And in the virtual lab, so there's actually an AI professor agent. And working with the AI professor agent is actually a bunch of different like AI student agents, right? Each of them will have different expertise, maybe in like computer science or computational biology or data science or immunology. So these AI agents, actually, they run their own group meetings. And they also would give them a physical budget so they can actually do some experiments. And we also created like a school, a virtual lab school, where the agents can actually go to the school and then to teach themselves how to become a better and deeper expert in one of the domains. So I'll explain a bit more of how this virtual lab works and then share the kind of innovations and research they're able to do. So first, I'll just explain how we interact with the virtual lab agents. right? So the human researchers, we interact with the AI agents. Mostly I can do this by talking to the AI PI or the AI professor agent. So for example, I can tell and explain to the AI PI some description of like, what is the project that I'm interested in, right? Usually it would be a high level description. So for example, I can say, oh, I want to design like a new kind of protein, right? Or I want to analyze this very complex genomic data set, right? And then it's the job of the PI agent then to say, okay, given that project description, what are the different areas of expertise that are needed to tackle that project, right? So maybe for this particular project, the PI agent would say, oh, it's useful to have like a computational biologist on the team, right? In that case, then the PI agent then would train and to create a sub-agent that have this expertise in computational biology. For different projects, right, different areas of expertise might be needed. So then the PI agent has the flexibility then to recruit and to create and spawn different agents with those different expertise. I'll come back to you in a couple of slides to explain more about how that training process of agents looks like. But for now, you can think of each of these agents as being like starting off from like a frontier language model, could be GPT-4 or GPT-5 models, right? And then we do additional fine tuning on top of these models, and additional, then we also enable these models to use these domain-specific tools to enable these models to become a better researcher and a better expert. So after all these agents are created by the PI agent, right, so then they can actually run team meetings or group meetings. So in a group meeting, then all of the agents will actually meet together. So similar to our human meetings, the different agents can contribute ideas, and then they can come to a consensus about the research plan for the project. In addition to these group meetings, the agents can also run one-on-one meetings. That's where one of the agents might meet with the PI agent, and then they can solve like a specific subtask, like maybe reviewing a piece of code or analyzing a particular data set. Right? And all of those individual works of the agents would happen in sort of like a virtual sandbox environment where the agents are able to use different tools like Jupyter Notebooks and AlphaFold and other tools. Okay. So just to make this more concrete, so here I'm showing an example of what one of these virtual lab agent meetings will look like. So in this project, so this is actually the first project we gave the virtual lab, where we basically told them that, okay, we want to design binders to the recent COVID variants. And at the time of this project, right, there were actually not good binders to the recent COVID variants. Right? So we thought, okay, this is kind of actually a very challenging open-ended research project, but also a very timely public health project for the agents to try to help us to tackle. So you can see that in the first part of the meeting, right? So the PI agent will then call the meeting together with all these other agents, then first explains to the other agents, like, what is the objective here, right? So it explains that, okay, our job here is to design binders to the spike protein of the newest variant of SARS-CoV-2, right? And also explains why this is an important problem. And after that, the different agents can now speak up one after another sequentially to contribute their ideas. In this case, the immunologist agent spoke up next, and sort of actually made a kind of interesting suggestion of designing these binders in the form of nanobodies. Now, this is already kind of a surprising recommendation, because if you ask human researchers, most of the time you think about designing binders in the form of what's called antibodies, which are much more common. It's often used as sort of substrates for like vaccines or drugs. Now, nanobodies are sort of small cousins of antibodies. They're only found in a few animals like camels. Right, so it's kind of an orthodox recommendation to design nanobodies rather than the more common antibodies. But as we'll see shortly, this actually turns out to be quite a wise suggestion. And the reason for this is that we'll see actually is when the machine learning agent spoke up next, right? So the machine learning agent says, okay, I actually like this idea of going with nanobodies rather than antibodies. And the reason is that these nanobodies, because they're smaller, that actually makes my job as the machine learning agent easier, right, to computationally model the structures of these nanobodies because they're smaller proteins. And then recommends using these deep learning models to do that. And then the computational biologist agent spoke up next and says that, okay, I also recommend using some more physics-style molecular simulations to study the structures of these proteins, right, in addition to using deep learning models. And in all of these virtual lab meetings, we also have what we call a scientific critic agent. So sort of like a reviewer agent that sits in on all of these discussions, whose job is to provide more skeptical feedback and also try to poke at holes at the discussions of other agents. But in this case, the reviewer, the critic agent says that, well, if we're going to go down this path of developing nanobodies, since there's not a lot of public data around nanobodies for COVID, then you need to be super careful about overfitting these different models to these nanobodies.
So, Professor Zhao, there is a question that what are
Yes.
you using for fine tuning? What specific data and the training approach you are using?
Yes, it's a good question. So I'll explain the fine-tuning part as a part of the agent school in 2 slides. But basically, we're actually having the agents essentially retrieve and download relevant papers themselves, right? So the agents actually find the relevant papers by doing like PubMed searches or also finding textbooks, and then they can do fine-tuning on top of those relevant literature. Great. So I hope this gives you a flavor of the kind of agent meetings. So the actual meeting goes on for longer. So here it's basically just showing the transcript from the first half of the meeting. But you can already see how different agents can build ideas off each other, right? And come to a consensus about how to tackle this quite challenging and open-ended research project. One thing that I'm very envious of the agents is that their meetings are much more efficient than my meetings. So one of their meetings maybe would take a few seconds or a minute. So in the time that I had one of my meetings, right, so the agents actually already had hundreds of research discussions. And because they don't get tired, right, these agents, they actually run multiple research discussions in parallel. So basically for every scientific topic that they want to discuss, they will actually discuss it 5 times in parallel in 5 parallel meetings, right? So in each of these parallel meetings, they'll be discussing the same topics, but in different parallel meetings, right, different agents might speak in different orders. There's also some randomness in the base language models. So they often come up with different ideas in these parallel meetings. So it's kind of an effective way to try to capture more creative ideas. And then what we do is we ask the PI agent to read the transcripts of all these parallel meetings and then come to a consensus of what to do next. Right. So by running these meetings in parallel, then we enable the agents to be a bit more creative and to explore a broader range of ideas. And in case you're curious, oh yes, please.
Yeah, sorry, like there was 2 questions also on this, that how does this deal with the holes in biological knowledge? Existing knowledge of nanobodies is incomplete.
That's right, yes. So that's also a good question. And basically what the agents end up doing here is that they decided to create a new computational workflow to simulate and to basically study the nanobody structures in silico. Right? So that actually enables the agents to fill in gaps in the literature by doing their own in silico experiments and models. So I'll explain this in silico workflow that's created by the agents. Yes.
Yeah, maybe like you can continue your thing, and then after a point, like we can stop and take other questions. There are a lot of questions coming in.
Okay, yeah, sounds good. Sounds good. I'll pause shortly. Yeah.
Yeah. Okay.
And here what I'm showing in this pie chart is basically how often the different participants will speak in these virtual lab discussions. All right, so for example, the human researchers, which could be myself, my students, maybe we speak about 1% of the time. So we don't have to talk very much. Right? So our job is to mostly provide high-level guidance to the AI agents. So for example, we would explain to the AI agents, like we want to, what projects are important to us as humans, right? And we also explain to the AI agents what are some of the constraints we have. For example, like here's how much money we have to do the wet lab experiments, so they can't go too crazy in their designs. But otherwise, we mostly we don't want to micromanage the AI agents. We mostly leave it up to them to come up with their own research plans and to implement and execute on their research ideas. Right. So our job is to mostly provide this high-level advice and guidance. In this particular project, the computational biology agent actually does the most speaking, right? Which I think makes sense because here the computational biologist agent has to do a lot of the work in actually coming up with these computational pipelines and try to design the nanobodies. The machine learning agent's job is actually to basically to write the code for the other agents, right? So for example, other agents would tell the machine learning agent, right, implement this algorithm, right? And then the ML agent then will actually write the code in Python to implement those algorithms, right? So it doesn't have to talk very much in English, but actually does all the coding for all the other agents. So now that we've seen a bit of how the virtual lab interaction happens, I want to come back to this question about how do we actually train the agents to the agent school. And this also answers some of the questions that the audience have asked. So we created sort of almost like a replica of Stanford, where the agents can actually go to their own school and then teach themselves how to become a better expert. So the reason for this is that since these agents are built on top of these large language models, right? If you take all of these off-the-shelf language models like GPT-5 or Anthropic models, right? These models are quite good at general broad knowledge. But if you go really deep into a particular scientific domain, then these models may not have the most, like the deepest understandings or the most up-to-date knowledge for that particular domain. So filling that gap, that's where we decided that we need to have a school where the agents can actually go to the school and then try to learn the extra bit of knowledge to become really an expert in that particular domain. So here's how the school works for the agents. So first, we would tell the agents, here are some of the topics that are important for them to learn about, right? For example, one topic could be the agents should learn about like the latest developments, right, in the different COVID variants. Or maybe the agents should learn about like the latest computational tools like AlphaFold. And then for each of these topics, then the agents will actually generate their own learning curriculum, right? So in the forms of they would come up with a set of questions that are relevant for each of these topics. And then based on those questions, we allow the agents to basically do searches to internet and through other resources to basically find and retrieve papers and articles that are relevant for each of these topics. So for example, the agents can do a PubMed search to find open access biomedical papers that are relevant for their topic. And then they would download those papers and do a deep read of each of these papers that they download. As the agents are reading these papers, then we allow the agents to basically update their model parameters. So basically what happens under the hood is that each of these agent is a large language model, so it has a transformer neural network. So as they're reading these papers, then we allow the agents to do supervised fine-tuning to update the weights of these transformer neural networks that help them to ingest these additional new insights and new knowledge. So even when the agents are starting off, maybe they all start off as being the same transformer, right? It's initially the same language model, right? Like GPT-4, right? But as they read and go through the school, then it starts to specialize, right? And the parameters of these models starts to diverge, right? So they become different, they start to develop different expertise areas. And it will not be a very good school if there's not some sort of assessment mechanism. So basically, after the agents have done their readings and done their model updates, then we have like a bunch of tests that they have to take. So these tests are actually designed by other agents, by sort of teacher agents, right, who come up with these questions and QAs, quizzes, and then the student agents will have to pass those quizzes before they're allowed to graduate. Right? If they don't do well on these quizzes, they have to go back to school and find additional papers and do additional model updates. So this whole school process is actually quite autonomous, right? So the only human input is basically what topics they should learn about. But otherwise, we allow the agents to basically find their own, design their own learning curriculums, find their own materials, and then do their supervised fine-tuning to update their model parameters. And the benefit of this is that because it's quite autonomous, right? So this enables the agents to learn about a lot of new topics beyond what we have expertise to teach the agents. As for example, I don't know too much about the recent COVID variants. So then the agents can actually learn about those topics beyond what I could teach them. So maybe I'll pause here to see if people have any questions about how the virtual lab interaction looks like and also how the agent school works.
Yeah, thank you, Professor. There is a question from Garima Rani, how to validate these meeting outcomes and be sure it is not just made up.
Yeah, so it's a good question. So there are a few ways we should validate these meetings, right? So first is that ultimately the agents have to write some code, right? And they have to use these different tools. And we can validate these by seeing like, do this code actually have any bugs in them, right? And do they actually do what they're supposed to do, right? So we can basically run unit tests, and actually the critic agent, their job is to actually try to debug this code and then make sure that the code actually works as expected. The second is that ultimately, like, the final ground truth comes from And final validation comes from doing a wet lab experiment. Right? So the agents would actually do their in silico work and design these nanobodies. And then I'll come to in a couple of slides of how we then actually will physically make these nanobodies and test them in the real world to provide sort of the ultimate validation to see do these actually work as it intended as good binders to the recent COVID variants.
Okay, thank you. There is one question from Atik Bhatti. How was the quality of articles determined by the agents? Whether that was a quality study that used as a reference?
Yeah, so that's a little good question. So here we just sort of ask the agents to select their own articles. So first they would do like a broad, let's say a PubMed or Google Scholar search, right? They'll find a lot of papers that come up through PubMed as being maybe relevant for that particular topic, right? Maybe they can have hundreds of papers, right? And then we ask basically the agents to first read through the abstract of each paper and find which papers maybe are the most salient and most relevant for their topic. Right, then maybe from those hundreds of papers, they'll come up with like a list of, let's say, 50 papers that are the most relevant. And then we allow them to basically download those papers and then read each of those papers in more detail.
Thank you. There is a question. Will professor be available to talk about refining this research in this field out of the lecture? If yes, how to approach him?
Yes, I think probably the best way is that you can email me and my email is easy to find on it's just jamesc@stanford.edu.
Okay, there are actually a lot of questions. You want to take all of them?
Maybe we can take one more if that's okay, and then we can
Okay.
and then I'll pause again to take more questions later.
Yeah. So there is one question. How do we make context cleaner? The input requirement assume clean data, but in reality, data is often messy.
Yeah. So here it is quite messy information, right? Because for example, in the agent school, they have to read all these papers and there's a lot of potential noise or conflicting information or even contradictory information in those papers. Right. So this is where we actually mostly leverage the reasoning capabilities of these language models to sort of resolve and to reason through these conflicts. And the current models, especially like the GPT-4 or the GPT-5 or Anthropic models, actually, in our experiments, we found them to be actually quite good at doing these reasoning and try to reason through some of these ambiguities.
Thank you. You can move. Again, you
Okay.
can ask me to stop and I can ask the
Okay.
questions.
Sounds good. And so we'll stop every few slides periodically. So great questions. So now that we've seen how the agents interact and how they're trained, then I want to come back to this case study, right, of to illustrate what are the kinds of science that they're able to do. Right, so recall that the first project that we give to the virtual lab was we asked them to design binders, right, for the recent COVID variants. And the agents decided to do this in the form of designing nanobodies. Now, because there's not a lot of public data on nanobodies for COVID, the agents decided that they wanted to create a computational workflow to help them to model and study the properties of these nanobodies in silico. So here's the workflow that's created by the agents. I think it's actually quite fascinating what they did. So they first decided to use these different tools to help them to look for promising candidate nanobodies. So first, the agents decided to use this tool called ESM, which is basically a protein language model. Right. So what it does is that it takes ESM, will take its input, some amino acid sequence, and just computes a score to see how likely is that sequence to form like a stable and functional protein. Right. So this will be sort of like a first filter for the agents to decide, okay, so these are likely potential candidate nanobodies. And then the agents will feed those nanobody candidates right, into AlphaFold together with the spike protein of the target COVID variant. What AlphaFold does is that it creates this three-dimensional structure of how the candidate nanobody is slightly likely to bind to the target spike protein. The agents will actually look at that three-dimensional structure and then pass that to a physics tool called Rosetta, which does more physics-style molecular simulations to relax that structure and then to refold it to estimate the binding energy. So each of these different tools provides kind of a different and complementary information to the agents, right? So the agents actually will look at the output from all of these different tools to come to like a holistic score to say how likely is this candidate nanobody to be a good binder. And then they would iterate through this process multiple rounds to come up with the best candidates. And this workflow is actually, to the best of our knowledge, different from what people have published before. Right. So previously, people maybe have used different tools, but this is sort of, in our view, from what we've seen, sort of the first time that people that there's this workflow that actually uses, this particular set of tools for optimizing these nanobody designs. So this is kind of interesting. And this computational workflow itself actually ends up being super useful because the space of nanobodies is actually just astronomically large. There's trillions upon trillions of potential nanobodies. But by running it through this computational tool, the agents are able to relatively quickly came up with like a list of a much smaller list, about 92 nanobodies. And the agents thought, okay, you just have to test these 92 rather than these trillions of potential candidates, right? And these 92, the agents were pretty confident would actually have some pretty strong binders. And now the agents, they don't do their own wet lab experiments. Right? So basically the agent said, okay, so here's a set of nanobodies, the 92, that's the best candidates. They give that to us. And then we would actually work with our experimental collaborators to make these nanobodies in the real lab. So we synthesize them in the real lab, and then we test each of these nanobodies against the spike protein of the, to see how well do they bind to different COVID variants. So here I'm showing sort of two example nanobodies that were designed by the virtual lab agents. And the bottom here basically shows the experiments that we did to test how well do they bind in the real world. So I'll just explain what each of these curves mean. So if we look at the first curve, this bottom red curve was the open circles. That's basically if we take the existing nanobodies designed by human researchers, right, and test those against this recent COVID variant called JN1 variant, which started to spread last year. You can see the human designed nanobodies actually does not bind to this JN1 JN1 COVID variant. The second red curve was the solid red circles now. These are the new nanobodies designed by the virtual lab agents. They actually now see, they do show more promising binding to this JN1 variant, as we see in these experimental curves. And the two blue curves basically show if we take the same nanobodies and test them against a very different strain of COVID, the one from Wuhan 5 years ago. You can see that, again, the solid red curve, the solid blue curve is the one designed by the agent. They actually show even stronger binding to this Wuhan strain of virus compared to the nanobodies designed by the human experts. So we think this is actually quite exciting as the agent's able to design these nanobodies, right, relatively quickly, so just within a few days, right? And we can then do the experiments to validate that these nanobodies designed by agents, they actually seem to show pretty promising binding across quite different strains of COVID. We did additional experiments, which I'm not showing here, but we test these nanobodies like also against other viruses and also against human host proteins to show that these do not bind to these other viruses or against our own proteins, which is a nice property to make sure that these nanobodies are actually specific to COVID, right? So it reduces potential side effects. And now we've also given these figures here, right, these experimental results back to the virtual lab agents, that they've gotten that real world feedback and they've actually used that to further refine and improve their computational pipeline, right? So there's actually sort of an iterative process. So just to wrap up this part of the story, right? So we designed this virtual lab to be actually an open source platform. So it's already open source as a part of this paper that we put out so that people here in the audience can also use it. Right. And it's also designed to be quite versatile, right? So it's not limited to designing proteins, right? You can use them to tackle other kinds of research projects. It could be quite open-ended research projects, right? In statistics or data science or biology or even physics and other domains. And one experiment we did is that we found that when we have teams of multiple agents, each agent was a different expertise, right? That team often performs much better than if I just have a single agent that tries to solve the problem by itself. Basically, what happens is that when you have different agents with different expertise, right, when they have group meetings, so often they get into discussions or sometimes disagreements with each other. Right? And when the AI agents disagree with each other, they also have to resolve those disagreements. They're very polite when they do that, more polite than human researchers, but they still have to figure out how to convince each other. And that's actually when they get into some, I think, quite interesting and also more robust reasoning, right? When the agents have to debate and disagree and resolve their discussions. We also found it to be quite useful to provide the agents with sort of some memory and also like a sandbox, basically a virtual environment where they can safely run and execute their different tools. As you see here, the agent actually do a lot of work by like running tools like AlphaFold and other domain-specific tools, right? And it's useful for the agent to be able to run that safely in like a sort of like a Docker container or like a sandbox environment. And finally, I think the agents also have their own social dynamics, which is quite interesting. Right, so similar to in human research collaborations, right, some people talk more than others, right? In some group meetings, depends on who speaks first, right? That also changes the trajectory of the discourse. Similarly, when we have these teams of AI agents, right, depends on which agent speaks first. Sometimes agents talk more than others, right? That also can change the dynamics. And one thing that's kind of nice is that we can study the social dynamics in agents much more easily than we can do that with humans, right? For example, because we run multiple meetings in parallel, in these different parallel meetings, we can actually specify which agents speaks in first, right? We can also even change the number of agents that participates in these group discussions. So we can see that, okay, if you have too many scientist agents, right, does that actually make the research better or worse? So I think sort of like a nice petri dish, do a lot of these experiments to figure out what is sort of the best organizational structure of scientists, right, that enables us to be the most creative, also to be the most effective. And maybe then we can take those learnings and also help to inform how we design our human research interactions. Okay, great. So I'll also pause here then to see if people have additional questions.
Wow, it's very, very interesting. Yes, there are a lot of questions. So one of the question came, were parallel meetings under the same conditions, or did the convos run under different settings, except different temperatures to get variability?
A good question. Yeah. So here we just run the different parallel meetings under the same conditions. Because we use a default temperature for these different language models, so that already produces enough randomness across the different parallel meetings.
Okay. And there was a question, how do we handle the context bloating? Do you use any memory management system? I guess you probably said that.
Yes.
Yeah.
Yes, that's right. Yeah. So basically what we do is that every agent has their own memory mechanism. So they remember what did they talk about and discuss from the previous group meetings and from their previous one-on-one meetings with the PI agents. So that actually helps the agent to sort of stay in context. And the other thing that we found useful is that basically the PI agent or the manager agent basically periodically would summarize the current status for the entire team, right? So say, okay, so here's what we've done so far, and here's the next to-dos. And also it's useful to update all of the agents and then so that everybody's on the same page.
And it's related to that how the speaking order of the agents can affect the outcome of the meeting.
Yeah. So I think this is quite interesting. So we do find that, for example, here we can actually use different kind base language models, right? As that they also have some different personalities. We can also even explicitly inject different personalities to the different agents, right? So that it maybe and it's useful sometimes, for example, for the critic agent to be a bit more skeptical, right? And then maybe for the PI agent to be a bit more optimistic. Right. So that's something that we're still actively investigating. Is basically what combination of personalities and personas for the agents actually leads to the most creative research.
Yeah, one question is about the cost. That given the variability in API cost, could you share a realistic cost range that might look like for running a virtual lab in a typical research project?
Yeah, that's a good question. So here, the virtual lab is actually relatively efficient. So that, for example, it took them maybe just a couple of days to come up and to implement this pretty sophisticated computational workflow, right? And that part would take, I would say, like less than $100 to do that, right? So the most expensive part is actually do the wet lab experimental validation, because we have to make these proteins in the real lab, and that takes a couple months to make them, and to do these binding assays. That would took many thousands of dollars. So it's much more expensive than the virtual lab computational part.
Okay, another question is, it is a single fine-tuned model for all agents, or each agent use the separate fine-tuned model?
Yes, each agent would be fine-tuned separately. because they have different expertise, so they actually have different learning curriculums, right? So different agents would have, find different papers based on their topics, right? So maybe the immunologist agent would read papers that are relevant for COVID, and the computational biologist agent would read papers that are relevant for computational tools. So since they have different papers they're reading, then they get fine-tuned separately and they end up being different models.
Okay, there is an interesting question. Another one, that are there instructions available for the other researchers to build the virtual lab?
Yes. So we have a paper that we published actually just a couple of weeks ago. And then also as a part of the paper, we also released all the virtual lab open source. Right? So we have a GitHub page. The link is in the paper that basically provides instructions for how to use the virtual lab. Great.
Okay, another interesting
Maybe we
question.
can take one more
Yes,
question
okay.
and then I'll move on to the
Yeah,
second part. Yeah.
yes, exactly. I mean, there are flows of questions, I cannot even read all of them. But one thing was, yeah, with regards to the social dynamics, did you set any parameter? For example, always choose the cheapest or shortest time or the highest quality route to find the solution of the problem?
Yeah. We did not do that. We actually mostly left it up to the judgment of the PI agent to decide what to do, right? So the PI agent actually would often lead to the other agents, to give all the agents some time to talk a few times, right? A few rounds, and then try to reach a consensus of what to do next. Okay.
Yeah, you go ahead now. Yeah.
Okay, so yeah, great set of questions. So in the second part, I want to talk about also like a recent follow-up work that we did, specifically about building AI data scientist agents. And I think especially given this audience community, this might be particularly interesting. And here we built this agent that we call CellVoyager, which can take its input, ingest these quite complex different data sets as inputs, right? So a CellVoyager agent actually generates a bunch of different Jupyter notebooks in parallel, right? Each of these notebooks then will be sort of an interactive analysis that goes in depth of analyzing this data set. And in the end, it will generate sort of an analysis report, which includes these figures and interpretations of the figures to explain like what are sort of the new insights that the agent learned by analyzing this data set in depth. And the motivation for developing this kind of data scientist agent, especially for computational biology, is that modern biology is often faces with these really quite complex high throughput data sets, right? So for example, there's things like single cell sequencing data sets now that would contains many millions of cells, right? And also many thousands and tens of thousands of genes, so generate these huge matrices. And there are a lot of other kinds of data sets that captures things in protein levels or epigenetics and so on. And this is actually quite challenging for practitioners, for users, right? Because on the one hand, right, so these data sets are becoming larger and more complex. On the other hand, now there's just also a huge space of different computational tools, right? So it's just in the space of single cell analysis, now there's often thousands of different tools that one could use. And finally, there are just even many different kinds of thousands of different kinds of analysis one could do on top of those data sets, right? It could be clustering, differential expression, imputation, missing data. So a lot of things that one could do. It's often very challenging for like an end biology user to figure out what analysis to do, what tools to use, right? And how to really analyze such a huge data set comprehensively. And the result of this is that often people might not might miss some important biological insights, right? So for example, the end users may only be familiar with a subset of the tools, right? They may not have a lot of time and resources to really try a lot of different tools, right? And sometimes they may not think of certain hypotheses because they're less familiar with the literature. So we want to see, can we actually have an AI agent that can really help us, help the end users to fill in some of those gaps, right? And then to work with the users to explore the data set in depth and potentially to find some of these missing analysis. So the input into this agent then would be sort of some complex data sets and also maybe like a report or summary of what did people have previously done with this data set. Right? And then the output of the agent then will be sort of a new report, right, with figures and also reproducible code in the form of these Jupyter notebooks. right? That it contains sort of new insights that agent has learned from this data sets beyond what people have previously done, right? And then the users can also communicate with the agent interactively to further refine these analyses. So a little bit more about how this workflow CellVoyager agent looks like, right? So it has a few different steps, which I'll go through one by one, right? So first, the agent actually basically prepare the inputs, right? And it does this by looking at for given data sets, right? It loads the data sets, explore it a little bit to figure out what is in the data, right? And also reads what analysis have people previously done with this data set, which could come from previous papers or previous publications, right? And it also sets up the environment, right, with the Jupyter environment with all the corresponding analysis tools. And then the agent will actually do more like a deeper research, right, to find all the relevant papers and figure out what are the kinds of open questions in this field that might be relevant for this particular study, for this project. And then sort of sets up the Jupyter notebook with the data and imports the relevant packages. And after that, the agents come up with a set of hypotheses, right? Each hypothesis then will be investigated in depth in a Jupyter notebook, right? So basically what happens is that the agent will take one hypothesis, right? And then generates initially some initial code and run that code in the Jupyter notebook, which will generate some figures, right? And then it would interpret that figures to say, okay, so what can we do next, right? And then generate the next block of code, the next kind of follow-up analysis plan. Right? So we iterate through that process, multiple rounds in each of these Jupyter notebooks, right? And then it would analyze multiple analysis, multiple hypotheses in parallel across the different notebooks. So here's an example of the kind of workflow or the workbook that's generated by the agents, right? So each block of this Jupyter notebook produced by the agent actually has a relatively structured format, right? First, the agent will say, what is the description of the analysis that it's trying to do? And then it would write the code to perform that analysis. It would implement that code, right, and to generate the figure, and then it would interpret that figure to come up with sort of the next analysis to do. So it iterates through that process and to get deeper and deeper interactions, deeper insights into the data set. So the way that one way that we evaluated this data scientist agent is that we actually give this agent some interesting previous papers, right? And we ask the agent to basically reanalyze those papers and reanalyze the data in those papers to come up with new insights that are different and might have been missed by the original authors. And the way that we verify this is to basically we actually give the reports generated by the agent back to the original authors of each of these papers, and then have the authors to confirm whether this analysis is interesting and useful, and also whether it's different from what they had thought of before. So I'll just walk through a couple of case studies to illustrate how what the agents did. So the first case study is that we looked at the paper that originally studied the single cell sequencing on patients with COVID-19. And the original goal of this paper was to basically study how different kinds of immune cell types are expanded or depleted as a result of the COVID infections. So here, CellVoyager agent generated 5 new analyses, and all 5 analyses were actually judged by the authors to be quite distinct from their own analysis in the paper, and also quite interesting. Here's one example kind of analysis done by the agent. Right, so here the agents autonomously generate these different figures. So initially, the agents generate a bunch of figures that would do like data exploration and data visualization. And here the agents actually relatively quickly found that there was a pretty interesting pattern here, whereby if I look at what's so-called pyroptosis score, which basically measures that the tendency of different cells to undergo inflammatory cell death, there's a pretty strong difference in this pyroptosis score after in the CD8 T cells in patients with COVID infection versus the healthy controls, right? So which suggests that these CD8 T cells are after infection, and they're much more likely to undergo inflammatory cell death, right? And this is somehow specific for CD8 T cells and less seen in these other cell types like these CD14 monocytes. So this is actually kind of an interesting finding, so that when we give this finding back to the original authors, they thought, oh, this is pretty interesting. This is, they hadn't thought of this before because they didn't know about these pyroptosis scores. But in retrospect, this actually made a lot of sense to them, and it's also further confirmed by subsequent literature. In the second case study, we asked the agents to basically analyze a brain aging data set. So here, what the original authors did is basically they sequenced cells from the mouse brain across different ages. And the original study focused on building these machine learning models, right, to predict the age of individual cells. And here, the CellVoyager also performed 5 distinct analyses, and 4 out of these 5 were judged by the authors to be quite interesting. So an example analysis is as follows, where the agent also did some exploration and visualization of the data to look at what are different clusters of different cells. And then the agents actually found this pretty interesting pattern of how transcriptional entropy is actually highly correlated with the biological age of the cell. So basically, high transcriptional entropy just means that there's higher transcriptional noise in each of these cells, and it seems like cells that are biologically older tends to have higher transcriptional entropy. Right? So there's a pretty consistent pattern across different cell types in this particular part of the brain called the subventricular zone of the brain. And what's interesting here is that the similar pattern of correlation between transcriptional entropy and age has been reported previously in other tissues and other organs. But this is sort of the first time that people have shown this for this particular part of the brain. Right, so it also illustrates that the agent is able to do something that's not terribly surprising, but it is kind of interesting, and it's also a new finding that also contributes to the literature. So in this part here, so I think that this kind of data science is also a good use case, right, of these AI agents, in particular because the agents are quite good at summarizing the relevant literature, coming up with hypotheses, and also they're quite good at writing code, right? In this case, we enforce the agents to write all the code in the Jupyter notebook to make the whole process easy to reproduce and very transparent for the human researchers. Because then the human researchers can just rerun the Jupyter notebook to make sure that the agents did something that's reasonable. This also enables sort of a kind of human-in-the-loop interactive data analysis, right? So the human researcher can also tell the agents, I want to try this, analyze this other hypothesis, and the agent can adapt and then regenerate the code. So this part of the work is led by Sam Albert and Bowen Chen, who are 2 excellent PhD students working with me at Stanford. I'll also pause here for a bit and see if people have any questions about this part.
Yeah, I think there is a question that how does the AI develop and work on their own and have a proper coordination among themselves in the school and back end of AI schoolwork, such as agent can work independently?
Yeah, so the nice thing is that these agents, they're all, since they all are language models, they all communicate with each other and also with us through natural language, right? So they basically do all of their coordination through human language. So for example, the PI agent then would tell one of the sub-agents, okay, you do this part, right? Or in this case of the CellVoyager, there's also one agent that's basically interpreting the figures, right? So it actually provides the interpretation of the figure in the form of a paragraph of English, right? And then the other agent would look at that interpretation and say, okay, that makes sense. And then here's the sort of next analysis or next hypothesis based on that interpretation. So the coordination, it's actually pretty natural because the agents are able to communicate well through human language.
Okay, thanks. Do they develop new ideas on their own, or just give me 1 second. Yeah, or they just build whatever the paper is mentioning. I mean, on the ideas where the paper they read. Yeah.
Yeah, it's a good question. So here, the agents, they were actually able to find new ideas and new hypotheses that was not in the original studies in the papers. For example, the original study did not actually look at transcriptional entropy, right? And did not look at how entropy is correlated with age. And the agents actually read some other paper in other organs that said, oh, entropy might correlate with age, and thought, okay, maybe it would be interesting to test that in the brain, right? And then it tested that and found that, oh, okay, so there's indeed a kind of a strong correlation between transcriptional entropy and age in the brain. So I would say this is kind of like a hybrid. It's not an entirely new idea in that this has been shown in other organs, But this has not been shown in the brain before, or in this part of the brain before, right? So that the agent's able to take this idea from other discipline, apply it to this particular case, and find like a new insight, which I think is actually reminiscent of how a lot of human science has been done, right? People take some interesting ideas, maybe from one discipline, or maybe like a technique, a method from one discipline, and then apply that to a new problem.
Yeah, I think on that, actually, there is one more question that it may be possible that this similarity with the tissue in the last result is due to the biased behavior of agents, because sometimes this AI tool try to pick up conclusion from other system. So just to clarify this, how can we sure of this, or should we avoid this,
Yeah. So here it's a good question. So here, I guess the agent did try to say, okay, if you really stratify by all the different cell types, and it saw that, there's actually a quite consistent and statistically robust correlation. And so it generated these p-values, the agent generated these p-values to say, okay, across all these different tissues, there's actually a consistent trend where older cells tend to have higher transcriptional entropy. Now, to further validate this, I think ideally the agent should be able to like find other data sets, right, from other studies and then reanalyze that to further confirm this finding. So I think that would be a good next step.
Maybe I just take one more question and then you probably
Yeah.
can carry on, because
Sounds
a lot
good.
of questions. Okay. So um, can CellVoyager accommodate Bayesian analysis, that is combine analysis from the paper and the other analysis or findings on the other data sets or paper? I guess you already answered,
Yeah.
but not as a Bayesian perspective.
Yeah, I think actually Bayesian analysis is actually particularly suited for these agents because they could actually generate quite informative priors. Because these large language models, since they've read so much literature, they've read a lot more papers than I have, right? So they could actually generate a pretty informative prior about like what is the, maybe the distributions, and then use that prior then to do posterior calculations. I think actually Bayesian analysis is particularly well suited for these language models.
You go ahead.
Great. Yeah, thank you. So I also want to end by sort of discussing, I think, some important limitations of these AI agents that are useful to highlight. Right? And because this also highlights why we think of these agents as really being like collaborators and complementary to humans rather than replacements of human researchers. Right? So one limitation of AI agents currently is that I think they're currently better at using existing tools rather than creating and inventing entirely new tools from scratch. As for example, the AI agents, they will actually use AlphaFold as a tool and apply that to new proteins, but they're better at doing that rather than like creating a new version of AlphaFold from scratch themselves. And I think this is quite complementary to a lot of human researchers in that we are better at developing new methods, new tools, and the agents can then help us to apply those new methods, right, to new data sets and new problems, or be like the interface between new methods that are being developed and also the downstream end users. Similarly, I think the current agents are better and more at breadth, right, rather than depth, right? They read a lot of literature, but they don't go very deep into a particular domain. Whereas the human researchers, I think we often do go deeper into a particular domain, right? And we can try to be more creative within that domain. So I think that's also quite complementary. AI agents, they make mistakes. And in our experience, often these mistakes happen because the models do not understand the full context of the problem. Right? For example, if I give it a data set to analyze, but I don't tell the agents how the data set has been pre-processed, then the agents might make assumptions about missing values or different parts of the data that are not valid. Right? So that's why it's important for us to try to capture the metadata as much as possible in a really like an agent-friendly form, so the agent can really understand what is the broader context of this data, and also to provide humans to provide critical reviews at this important stages of this kind of data analysis. And finally, the current agents are still mostly limited to do in silico computational work, which is why collaborations with human experimental validation is still critical. I'll just wrap up by actually highlighting this, sharing this sort of experimental conference that we're running, in case folks here are interested. So we're running this new conference where for the first time, both the authors and also the reviewers of the conference are all AI agents, right? So you could have your AI agent, right, basically do the research and write the paper. The humans can be co-authors, right? But the AI agent should be do most of the work, right? And then submit that to our conference. And then each of the papers will also be reviewed by other AI language model reviewers. And we'll also have a team of human experts do the final selection and the final assessment. So and all the submissions and also reviews will be publicly available. So I think this is kind of an interesting experiment where we can really see like, what are the strengths and limitations of AI, both as authors and also as reviewers, right? And so I would be very interested to see the outcome of these experiments. I also encourage people here in the community to have your AI agents, right, do some research and then submit that to our conference. And we'll also have the conference itself, which will be October this year, where the different AI agents will be presenting this research. We'll be streaming that, making that all freely available as well. Yeah, so I'll just wrap up here. So I hope to show these couple examples to illustrate why we're quite excited about the potential of AI agents is really our partner, right, in biomedical discovery. So in the virtual lab, we've seen how these teams of agents with different backgrounds can make new innovations and tackle open-ended projects. And in CellVoyager, we've also seen how data scientist agents can help us to reanalyze complex data sets to discover new insights. And I want to, again, acknowledge my fantastic students and group that led these different projects. So thank you, and happy to take further questions.
Thanks a lot. It is like really, really eye-opening, like research you have described to us. But like, yeah, there are some questions, I have a lot of questions. So what are the methods available for building confidence in the agent's output besides human validation? How does it rank the hypothesis being explored, since there could be many that could be analyzed?
Yeah, so that's also a good question. I think one thing is that's there's interesting works about having the agents to basically elicit confidence from the agents themselves, from the language models themselves. So this can be done either by, for example, looking at the probabilities, right, the agents assigned to different words and for different responses. And then there's also interesting works of basically essentially combining that with techniques like conformal inference to generate valid and robust calibrated confidence, right, to quantify the uncertainty around the different agents' responses.
Can you update these agents with the new GPTs when they're released, or they're always set to the GPT 4.0?
Yes, so these in the virtual lab and also in CellVoyager, the systems are all compatible with different language models. So they're also compatible with GPT-5 or Anthropic cloud models or Gemini models.
Maybe one more question, is there a reason to use Python as an environment, because it is an interpreter type language, so the agents can immediately examine where they're going wrong?
Yes, yeah. So I think that's one benefit of Python is that it's quite interactive. And it's also, we liked the idea of having the agent, for example, produce these Jupyter notebooks, because that also makes it very easy and convenient for human researchers, because then we can just go back and then run different cells of the Jupyter notebook and modify things to basically reproduce the agent's discoveries and findings.
I still want to take one question, because I think this is important, that for the conference you mentioned that the papers for this conference should also have AI, or is it can be from some other specific area?
Yeah, so the topic of the paper can be basically any scientific topic, right? Could be like in chemistry or physics or statistics, right? Mathematics even. The only thing that we require is that the work should be primarily done by the AI agent, right? So the AI agent, you think of basically being the first author of each of these papers, and then humans can be co-authors, but mostly as advisors.
Yeah, I think, I cannot even read how many questions we have. We still have like 30 open questions, Professor Zhao. So you can imagine how popular it is, but we are on top of the hour. So I just have one question, if you guys, if you can send us the slides, we can also circulate that to the people who have registered for this webinar. That will be great.
Yes,
And
happy to
besides
send
that,
the slides.
thank you. And besides that, I would like to thank, on behalf of IISA, that, coming here and providing really eye-opening concepts that which can take us to the future research. So really, really thankful to you, and also to all the participants who joined and, engaged in the discussions.
Well,
Thanks
thank
a
you
lot.
very much. Yeah, I really enjoyed it. And thank you very much to all the organizers.
Thank you. Thank you all. Bye.
Bye.